Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome back. Today we're tackling one of the really big, persistent questions in medicine. Why, after decades of incredible progress, can't we completely cure HIV?
It's a fascinating puzzle. We have these amazing drugs, antiretroviral therapies, or RT, that work incredibly well. They can push the virus down to levels where it's undetectable in the blood.
Right, people can live long, healthy lives.
Yeah.
But there's always that but. Stop the treatment.
And the virus almost always comes back, usually, within just a few weeks. It's because of what scientists call the latent reservoir.
A hidden population of infected cells that just waits. And for years, the assumption was, well, the virus just sort of randomly goes dormant in some cells, passive hiding.
Exactly, like it just happened to find a quiet cell and bunkered down. But some really groundbreaking work, primarily from researchers at Case Western Reserve University, challenges that whole idea.
And that's what we're digging into today. This isn't passive hiding at all, is it? It's something much more active.
It really is. It's more like the virus is a master strategist. It actively forces the host immune cell, its target, into becoming the perfect invisible hiding place. It basically reprograms the cell for its own survival.
Okay, so let's break that down. The main target, the place where this reservoir forms, is a specific type of immune cell.
Yes, primarily these are quiescent memory CD4 plus T cells. And that word quiescent is absolutely key here.
Quiescent meaning resting, dormant.
Precisely. Resting, inactive. Think of T cells like soldiers in your immune army. They're either fighting or they're resting, waiting for the signal to fight again.
And our drugs, the RT, they mainly target the active soldiers, the ones replicating the virus.
That's right. Both the drugs and our own immune system defenses are geared towards cells that are actively churning out virus particles. If a cell is quiescent, resting,
it's flying under the radar. The viral genes inside it, the provirus, aren't being actively read out. It's transcriptionally silent.
Exactly. Invisible. But here's the twist from the new research. HIV doesn't just find a cell that's already quiet. It seems to actively make the cell quiet after infecting it.
How did the researchers figure that out? What tools did they use?
They use some pretty sophisticated methods, things like single-cell RNA sequencing, which lets them look at the gene activity inside thousands of individual cells simultaneously right after infection happens in these specialized lab models of T cells.
So they got a snapshot of the very early moments. What did they see?
They saw something quite dramatic. The HIV infection wasn't just causing a little bit of cellular stress, it triggered this huge shift, a massive reprogramming of the cell's gene activity, leading to a really strong, a transcriptional shutdown.
A shutdown. So the cell itself starts turning off its own genes. But how does the virus make that happen? It must be pulling some specific levers inside the cell.
That's exactly what they found. It's actively manipulating at least two key pathways in the host cell to force it into this quiescent state.
Okay, let's unpack those. What's the first pathway?
The first one involves a very well-known cellular pathway called P53.
P53. Isn't that usually called the guardian of the genome? It's involved in detecting DNA damage and stopping cells from becoming cancerous, right?
Yes, that's its day job, so to speak. It responds to cellular stress, pauses the cell cycle for repairs, or can even trigger cell death if things are too bad. The researchers saw P53 signaling get activated pretty early after HIV infection.
But wait, why would the virus want to activate a defense mechanism like P53? Wouldn't that be counterproductive? Potentially harming the cell it just infected?
But that's the clever part of the hijacking. In this specific context, activating P53 does something else crucial. It slows down cell proliferation. It puts the brakes on the cell's ability to divide.
I see. So by triggering this stress response, the virus actually encourages the cell to stop dividing and enter that resting quiescent state more quickly.
Exactly. It co-opts the cell's own safety mechanism to create the perfect hiding environment. Slower proliferation helps establish that dormant state.
Okay, that's one lever. Activate P53 to slow things down. What's the second piece of this puzzle?
The second key player they identified is a transcription factor called cripple-like factor 2, or KLF2.
KLF2, what does that normally do in T cells?
KLF2 is basically the cell's natural on switch for quiescence. In healthy resting memory T cells, KLF2 levels are normally high. When a T cell gets activated to fight an infection, KLF2 levels drop.
What did HIV infection do to KLF2 levels?
It caused them to shoot up, significantly and quickly. They reached levels that were basically identical to those you see in naturally resting quiescent cells. It's like the virus found the master switch labeled "go to sleep" and just cranked it up.
So you've got P53 hitting the brakes and KLF2 being pushed way up to actively induce that resting state. What's the combined effect on the cell's engine? It's machinery for growth and division.
The combined effect is pretty dramatic. You see a major downregulation, a shutting off of the key pathways that drive T-cell proliferation. The big one they noted was MYC signaling.
MYC, that's like the gas pedal for T-cell activation and growth, isn't it?
Well, precisely. MYC drives the cell to grow, divide, be active. And they saw MYC signaling get shut down hard and fast, especially within the first 72 hours after infection.
And shutting down MYC is enough to make the cell look quiescent.
They found that downregulation of MYC alone was actually sufficient to mimic the whole genetic signature of a quiescent cell. And alongside that, they also saw another pathway, E2F signaling, go down, which also confirms reduced cell cycle activity.
So the picture is becoming clear. HIV gets in, integrates its genetic material, and then actively manipulates P53 and KLF2. This coordinated action shuts down MYC and other proliferative signals, forcing the cell into dormancy and silencing the virus's own genes.
Yes. The latent reservoir isn't just an accident of infection, it seems to be an actively constructed outcome, built by the virus itself.
Is there specific data linking this KLF2 and P53 activation directly to the virus becoming silent?
Absolutely. That's where the single-cell data was so powerful. They could look at individual cells and see which ones had both KLF2 and P53 signaling activated.
And in those specific cells?
In those cells, the amount of HIV genetic material being actively transcribed the viral activity was reduced by an average of over 75% compared to cells where MYC was still active.
75% reduction. That's a huge difference and really points to this mechanism being central. It's not just random noise.
It strongly suggests this is a specific, targeted strategy by the virus.
Okay, let's clarify something crucial. Does the virus start doing this reprogramming immediately upon entry, or does it need to fully set up shop first, like integrate its DNA?
That's a great question, and they tested it. It turns out the KLF2 increase, that go-to-sleep signal, is dependent on the virus successfully integrating its genetic code into the host cell's DNA.
How did they show that?
They used a drug called raltegravir. It's an integrase inhibitor, meaning it specifically blocks that step where the viral DNA gets stitched into the human genome.
And when they added raltegravir?
The increase in KLF2 levels was completely stopped. It didn't happen.
Got it. So the virus has to permanently install itself first. Then it starts manipulating the cell to go dormant as part of the establishment process.
Seems that way. Integration is a prerequisite for inducing this quiescent state.
Which brings up the next obvious question. Do we know which specific part of the virus, which viral protein perhaps, is the mastermind behind triggering this P53 and KLF2 manipulation?
That's still being fully mapped out, but they have some strong clues. They did experiments using a sort of crippled version of HIV.
Crippled how?
It was a mutant virus that couldn't make a functional Rev protein. This meant it could only produce very early viral proteins, specifically TAT and potentially NEF.
Okay, so a limited set of viral tools. And did this limited virus still manage to shut down the cell?
Yes, it did. Even without the full complement of late viral proteins, this mutant virus still induced that same quiescence program, activating P53 and KLF2 and shutting down MYC.
That really narrows it down then. It suggests it's likely one of those early proteins, TAT or NEF, that's responsible for initiating this anti-proliferative effect.
Exactly. The current evidence points quite strongly towards the TAT protein playing a key role here, which is interesting because TAT is usually famous for activating viral transcription.
So TAT might be playing a double game, sometimes activating the virus, other times helping shut down the host cell to create latency.
It appears it might have this dual function, yes. It's quite a sophisticated level of control.
This whole discovery, understanding that the virus actively builds its hiding place, it must have huge implications for how we try to cure HIV.
Absolutely. The study collaborators really emphasize this. We now have specific host pathways, KLF2 activation, P53 signaling that we know the virus hijacks. These become potential new targets for therapy.
So instead of just trying to wake the virus up randomly.
Right, the old shock and kill strategy, which, involves trying to reactivate all latent cells, hoping RT or the immune system can then clear them. It's often quite toxic and hasn't been terribly effective.
But this new knowledge suggests a different approach.
It shifts the focus. Maybe instead of just waking the sleeper agent, we can disrupt the process the virus uses to put the agent to sleep in the first place, or prevent it from ever going dormant.
Like preemptively blocking the KLF2 increase or the P53 activation that HIV triggers.
Precisely. Target the host mechanisms the virus needs to manipulate. And the researchers involved are not just talking about this, they are actively developing new therapeutic strategies based on this principle.
Trying to interfere with this host cell manipulation directly.
Yes, with the hope that you could either prevent the latent reservoir from forming, especially if you treat early, or perhaps destabilize the existing reservoir.
And this mechanism, this idea of a virus actively inducing dormancy in its host cell,
Yeah.
could this be happening with other persistent viruses too?
The researchers certainly think so. This strategy of manipulating host cell quiescence pathways might be a common tactic used by other viruses that establish long-term chronic infections.
Like which ones?
Well, think about viruses like herpes viruses, which cause lifelong infections, or maybe hepatitis C, or potentially others we don't even fully understand yet. They all need ways to persist in the body long-term.
So understanding how HIV does this could potentially open up new ways to tackle a whole range of other viral diseases by targeting similar sleep switches in our cells.
It's a definite possibility. It highlights the broader importance of this kind of fundamental research into virus-host interactions.
It really underscores how learning the basic biology, the molecular tricks these viruses use, is critical, especially when a virus can permanently integrate into our DNA and then actively control cell behavior. That has implications for future threats, too.
It absolutely does. Understanding these mechanisms is key not just for treatment, but for overall pandemic preparedness and anticipating how new viruses might behave.
Okay, so let's try to boil this down for everyone listening.
Yeah.
The big takeaway here is that HIV's ability to hide and persist, blocking a cure, isn't just bad luck or random chance.
Right. It's an active, orchestrated process. The virus hijacks the host T cell using pathways like KLF2 and P53 to effectively force the cell into a dormant state by shutting down key growth signals like MYC.
It builds its own invisibility cloak.
A very effective one. And that leads us to a final thought, something for you to consider. If we now understand that latency isn't just a bug, but an inherent feature of HIV infection, something the virus actively works to achieve.
How does that change the way we think about what a cure actually means? Does it make the idea of a functional cure, where the virus is controlled without drugs, seem more realistic? Or does it redouble the focus on achieving complete eradication, now that we know the blueprint the virus uses to hide?
Knowing that blueprint, as you say, is the first essential step. It might finally give us the tools to dismantle that hiding place for good.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.